Hi everyone, in this video we will be demonstrating a lab where we will look at how the rates of photosynthesis change under different experimental conditions. So to start out, we'll explain the setup of this experiment, and you can see here that we've got this large beaker filled with water, and we also have a thermometer in the water to measure the temperature. Now next what we've done is we have a piece of Alodia, and Alodia is just a genus of aquatic plants that actually lives below the water surface and extracts carbon dioxide from the water instead of directly from the atmosphere like most terrestrial plants do. And so we put this Alodia into a test tube and filled that test tube with sodium bicarbonate, which will serve as the carbon source for the plant. You can see in our setup that the test tube is closed with a 2-hole stopper assembly, and we have placed the test tube in our beaker so that the water covers the solution in the test tube. Now you see that we have rubber tubing coming out of the top of the stopper on our test tube that is attached to a pipette that's filled with water, and we are introducing a small air bubble into the pipette where it is attached to the rubber tubing. And we're going to do this by inserting a very fine needle of an empty syringe into the tubing, and we're injecting air until there's a visible meniscus in the graduated portion of the pipette. So to begin our experiment, we've let our beaker sit for a little bit so that the water is room temperature at around 20 or 21 degrees Celsius. And we have also turned on this desk light, which is serving as our light source, and it's positioned about 30 centimeters from our apparatus. We're starting off with a control run of the experiment, and we will be measuring the position of the air bubble in the pipette every minute for 20 minutes, and then we will calculate the rate of gas produced per minute. The next thing we are going to do is to manipulate different factors and see which factors affect the rate of photosynthesis. So the first variable we will be looking at is light intensity, and we will measure the rate of photosynthesis at high, medium, and low light intensities by positioning our lamp closer to the setup at high intensity and even further away from the setup at medium and low intensities, respectively. So moving on to the next part of our experiment, the second variable we will manipulate is light quality. And to do this, we are going to run 10-minute comparative tests with red, blue, and green colored filters while still keeping the light at a standard distance of 30 centimeters from our Alodia setup, and we're going to keep the temperature constant as well. Now the last variable we can manipulate in this experiment is temperature. And to go about manipulating temperature, we can use our beakers filled with warmer or cooler water. So hopefully you all understand the setup of this experiment, and now you have the opportunity to learn how these 3 treatments affect the rates of photosynthesis in plants.